Hello everyone, we checked what is cell differentiation. Let's see what is the plasma membrane. The plasma membrane or cell membrane or plasmalemma that envelops every eukaryotic cell consists of phospholipids, cholesterol and proteins with oligosaccharide chains covalently linked to many of the phospholipids and proteins. This limiting This limiting membrane functions as a selective barrier regulating the passage of materials in and out of the cell and facilitating the transport of specific molecule. Okay, so first things first. Plasma membrane, you can call it cell membrane. Plasma lemma consists of what? It consists of phospholipids, it consists of cholesterol, it consists of proteins. Along with it has oligosaccharide chains and that are covalently linked to many of the phospholipid and proteins. So we have a lipid bilayer. We'll study about this lipid bilayer and what can be the role of a membrane. To It is the one that monitors the in and out of movement of molecules and substances in and out of the cell. That's basically the role of membranes. So it also has some proteins present that facilitate the transport. We'll see what it exactly does. So one important role of the cell membrane is to keep constant the ion content of the cytoplasm, which differs from that of the extracellular fluid. So the important role is controlling and maintaining the ion concentration inside the cell, which is different than outside the cell, that is the extracellular matrix. Membrane proteins also perform a number of specific recognition and signaling functions, playing a key role in the interactions of the cell with the environment. So membrane proteins, as I said, are responsible for interacting outside anything outside of the cell. To They facilitate the movement along the membrane, etc. Although the plasma membrane defines the outer limit of the cell, a continuum exists between the interior of the cell and the extracellular macromolecule. Okay. Certain plasma membrane proteins, the integrins, are linked to both the cytoskeleton and extracellular matrix component and allow continuous exchange of influence in both directions between the cytoplasm and materials in the extracellular matrix. So, certain plasma membrane proteins, especially the integrins, they are linked both to the cytoskeleton and the extracellular matrix. So cytoskeleton is the part that is inside of the cell and extracellular matrix it is outside of the cell. And these are integrins allowed the continuous exchange, and it is responsible for exchange in both the directions. So something going into the cell or something going out of the cell. Integrins, these proteins are responsible. I think integrins is something that needs to be highlighted. Okay. Membranes range from 7.5 to 10 nanometer in thickness. And okay, so guys, I have this ideology about histology specifically, that when it shows numbers or you know size and shapes, anything that describes a size of a particular specimen, we are going to highlight it. We are going to try to keep this in mind, just so that we have an idea when we see this histological specimens in practical and we learn practical knowledge. So I also have a belief that a lot of histological professors in your university, sometimes in my university as well, they go on technical details of the size of focal length of microscope or something like that. So we are not going to miss any data as we're going to study everything in very very detail. We are not going to leave any loopholes. So we'll keep checking about what is the minute details for a particular specimen. So cell membrane thickness is 7.5 to membrane phospholipids are amphiotic, consisting of two non-polar hydrophobic, water repelling, long chains through this. We have phospholipids. We have the phospholipids in each half of the bilayer are different. For example, the well-studied membrane of red blood cell, phosphatid- phosphatidylcholine and sphingomyelin are more abundant in the outer half, while phosphatidylserine highlight the other two. Very complicated name though. Okay, some of the outer layer lipids known as glycolipids include oligosaccharide chains that extend outward from the cell surface and contribute to the delicate cell. We know what is glycocalyx, and what is glycocalyx consists. It is some outer layers lipid known as glycolipids include oligosaccharide chains that extend outward from the cell surface and contribute to delicate cell surface coating. So some glycolipids from the membrane just merge outwards and give a thin coating, and that coating is glycocalyx. What is glycocalyx? Is basically glycolipids. With the transmission microscope, hem, the cell membrane as well as cytoplasmic membrane may exhibit a trilamellar appearance after fixation in osmium tetroxide. Okay, I don't think that is important. Appearance after so we know what is fixation. We know now what is fixation. Fixation is one of the steps where we add a specimen to fixatives, and this fixative here is osmium tetroxide. We also studied in the previous chapter that this is one of the most common fixative, osmium tetroxide. uh, it gives trilamellar look. Why does it give a trilamellar appearance? I think we'll just remember about the trilamellar appearance here, because that can be asked sometimes. The cell membrane looks trilamellar.